Mammalian hearts have almost no ability to grow new heart muscle cells, called cardiomyocytes, after birth. Thus, dead tissue after an adult heart attack is not repaired with new cardiomyocytes. It is instead replaced with scar tissue that weakens the pumping power of the heart and often leads to heart failure. One promising strategy to remuscularize the injured heart is the direct cardiac reprogramming of heart fibroblast cells into cardiomyocytes. In a paper published in Circulation, University of Alabama at Birmingham researchers Yang Zhou, Ph.D., and Rui Liu, Ph.D., have identified TBX20 as the key missing transcription factor in existing cocktails for direct cardiac reprogramming of human fibroblasts. Adding TBX20 to the reprogramming cocktail MGT133, they report, promoted cardiac reprogramming and activated genes associated with cardiac contractility, maturation and a ventricular location of the heart muscle cell. Mechanistically, they found that TBX20 synergistically colocalizes with the MGT reprogramming factors at cardiac gene enhancers, causing robust activation of target genes. Our study highlights the undocumented role of TBX20 as a vital regulator of direct human cardiac reprogramming, Zhou said. Enhancing the efficiency and quality of direct cardiac reprogramming from human fibroblasts is a critical step in the clinical translation of this technology. An important area for future investigation, Zhou says, will be testing TBX20 in vivo for direct reprogramming. Current cocktails for direct reprogramming of human fibroblasts suffer from low efficiency and insufficient production of functional cardiomyocytes. The UAB researchers identified TBX20 as the most underexpressed factor when they compared cardiomyocytes induced from fibroblasts with a current cocktail versus functional cardiomyocytes. The addition of TBX20 promoted cardiac reprogramming, as seen in activation of cardiac genes related to sarcomere structure, ion channels and heart contractions. A sarcomere is the smallest functional unit of striated muscle. Human heart fibroblasts of the ventricle will be the primary target for evaluation of in vivo reprogramming. Zhou and colleagues showed that TBX20 was essential in reprogramming these fibroblasts to induced cardiomyocytes, suggesting a therapeutic potential for TBX20. In detail, TBX20 primarily activated genes at the late stage of reprogramming, enhancing calcium flux, contractility and mitochondrial function in the induced cardiomyocytes. Mitochondria are the energy source for heart muscle contractions. TBX20 appeared to help the mitochondria in the induced cardiomyocytes switch to an adult cardiomyocyte-like respiration. Mechanistically, the UAB researchers found that TBX20 was bound to and activated cardiac gene enhancers. An enhancer is a short regulatory element on DNA that can bind activator proteins to initiate transcription or increase the transcription of a particular gene target. Transcription is the production of messenger RNA copied from the DNA gene to encode a protein. Enhancers often function at a distance from its gene target. This enhancer binding by TBX20, Zhou and colleagues found, required all three of the MGT cocktail factors to help activate cardiac genes. TBX20 alone was not sufficient to induce cardiac cell fate conversion. Omission of any of the three MGT factors substantially reduced reprogramming efficiency. Together, our findings demonstrate that TBX20 requires intact MGT factors to promote direct reprogramming and activate cardiac genes, and they support the notion that direct cardiac reprogramming is a synergistically regulated process orchestrated by multiple factors, Zhou said. Single-cell RNA sequencing profiling identified two major clusters of cells after reprogramming with TBX20. One was reprogrammed induced cardiomyocytes, but the other group was only partially reprogrammed. The cluster of reprogrammed cardiomyocytes had gene expression profiles resembling ventricular cardiomyocytes. At UAB, Zhou is an assistant professor in the Department of Biomedical Engineering, and Liu is an assistant professor in the Department of Medicine Division of Hematology and Oncology. Support came from National Institutes of Health grants HL 153220, CA 259480 and HL 149137, and the American Heart Association Transformational Project Award 969529. Medicine is a department in the Marnix E. Heersink School of Medicine at UAB, and Biomedical Engineering is joint department of the UAB School of Engineering and the Heersink School of Medicine. Yawen Tang, Sajazan Ariel, Xiaoxiao Zheng, Xinyue Zhou, Vladimir G. Fast, Jianyi Zhang, Rui Liu and Yang Zhou. TBX20 improves contractility and mitochondrial function during direct human cardiac reprogramming. Circulation. 2022 doi. 10. 1161. CIRCULATIONAHA. 122. 059713 University of Alabama at Birmingham. TBX20 enhances reprogramming of heart fibroblasts into heart muscle cells. Direct reprogramming is a potential therapy for heart attack patients. Science Daily. Science Daily, the 21st of October 2022. ift.tt/pq2rdbn. University of Alabama at Birmingham. 2022, October 21st. TBX20 enhances reprogramming of heart fibroblasts into heart muscle cells. Direct reprogramming is a potential therapy for heart attack patients. Science Daily. Retrieved October 21, 2022 from ift.tt/pq2rdbn University of Alabama at Birmingham. TBX20 enhances reprogramming of heart fibroblasts into heart muscle cells. Direct reprogramming is a potential therapy for heart attack patients. Science Daily. https://ift. TT, PQ2RDBN. Accessed October 21, 2022. Thanks for watching. Please subscribe my channel for more videos.